Hello, I'm Robert, and my background is that for the past couple of years or so, I have actually been studying on this very topic of uh, planetary protection for NASA's Mars sample return. So, when I read NASA's draft environmental impact statement, I'd already read a lot of the literature and I knew the background, and I was astonished because NASA are normally so reliable, you don't expect this of them. They're normally well grounded, they normally have recent sites, they're normally up to date, but this wasn't up to NASA's standard at all. It was full of mistakes. And this is one of the most serious examples, and I actually alerted them of this with their first round of public comments in May on the ideas for the statement before they had the final statement. I said to them, are you aware of the ESF study from 2012 that said that the release of a single unsterilized particle larger than 0.05 microns is not acceptable under any circumstances? I explained this is to contain starvation amid ultramicrobacteria, and it's well beyond the capabilities of BSL-4 facilities. And they did not mention, they say in the draft EIS that they have read the public comments. They gave a section of responses to the public comments. They did not mention the ESF study from 2012. They gave no study reason for not including it. They didn't mention that it would be beyond BSL-4 capabilities. They gave no reason for questioning their suggestion to return it in a BSL-4 facility. They show no indication of having read my comment. And then this is an example, this is just a diagram I did to show this was the size limit through to 2012 before the study. This is what they reduced it to. NASA's study does not include the research that reduced the size limit from there to there. They are not aware and they do not mention this new size limit. And then this European Space Foundation from 2012 reduced that limit and said that reduced that limit, said that the next size limit review would be needed. That it need to be looked at again. Because there's a possibility it could be reduced further. And this is my own personal view, not their statement, that the next size limits review, there's always been an interest in these ribocells. A ribosome, not a microbacteria, can't be much smaller than that because, and the ESF said this is about the minimum size for terrestrial biology, because of the huge ribosomes, which are so big they pretty much can't even be smaller than that, because the ribosome is a enzyme that is essential for cell processes, and it is made up of a mixture of RNA and protein. The protein make it much more efficient, but they make it much bigger. The ribozyme, but it's impossible that when life started off that it had ribosomes. It can't be the very first thing produced in life because it's so complicated. And so there's the idea of a ribocell, which is a theoretical sized early form of life that would use ribozymes, and it would have no protein inside. It hasn't yet learned how to make protein. And instead, the idea is that it would use ribozymes, which are enzymes made of little fragments of RNA. They're much less efficient than the ribosome, but they would be able to replicate still, though more slowly. And there's increasing, way back in 2012, there was much less plausibility about this. There's been more research since then. Even in the last year or two, there's been new research on ribocells. It's a very active project of research, and I think a size limit review might well decide that they should contain hypothetical ribocells. If so, that is the diameter of it, the length of that line. And so I'll talk a bit more about that in a minute. So that's one example where the environmental impact statement, I mean, they just haven't considered the latest research. That surely is enough. If you imagine taking that to a court case, and justice says, what's your reason for not including the ESF sized study? Look, there was even this comment on May the 15th saying you should look at it. What's your reason? And the lawyer for the NASA would just say, I don't know, or something. And I think that it would be, at that point, they'd throw it out. And that would be, and the worst case remedy here is that the court would say to NASA, well, you can't return the sample. That's quite a lot of jeopardy in using an approach like this. I don't think they're aware of that. I don't think they have legal experts there. I don't think they have public experts. I don't think anyone's looked at this except engineers. And then it's just one of numerous examples, because if they at least doesn't show any sign of being looked at by experts who care about the effect of future legal cases on NASA. This is that's one of numerous examples. This is another example. They say in the draft environmental impact statement, existing credible evidence suggests the conditions on Mars have not been amenable to supporting life as we know it for millions of years. Now, by usual academic standards, you expect the thing in brackets after that to be supporting evidence, support that evidence. And you look at the most recent one, that's obviously the most important of those. And so you go and look and click through that because it's most recent. And then if you click through that, you expect this to be a site about Mars not having habitats for life. And you find a whole section about currently habitable environments. And you talk about habitats on Mars that are looking for established weather, localized habitable regions currently exist within these seemingly uninhabitable worlds, and that includes Mars. And so it's all about searching for currently habitable environments. Their site. The National Research Council from 2022. And yet they use that to support their claim that existing credible evidence says Mars hasn't been habitable for millions of years. Or is that supposed to make sense? So you can imagine them asking the NASA lawyer, and the NASA lawyer saying, oh, I don't know why they said that. And here's another example. First, potential Mars microbes would be expected to survive ejection forces and pressure. You expect the site to be about that, but it says explicitly they did not study microbes survival as a result of ejection forces and pressure. They didn't study sterilization at that stage. So the site explicitly said it didn't study the topic that NASA cited it for. Again, you think about, I don't know that. I'm not quite sure why they said that. I mean, that's not going to impress the justice. And by using all this, they conclude falsely that there's no real need to protect Earth from microbes from Mars. They're just doing it against an abundance of caution. That goes against all these Mars sample returns. It goes against their 2009 study that they use. And they used the 2009 Mars sample return study, the one they used for the BSL-4 sample return. And that said very clearly that the risk of a large-scale effects on Earth can't be ruled out. They said it's believed to be very small, but it's not demonstrably zero. And NASA came to the conclusion by misusing their sites that the result is not significant. There's no significant risk of large-scale effects. And they say that the environmental impact statement, directly contradicting the NASA Mars sample, the National Research Council Mars sample study from 2009. And not because they have better knowledge than them, but just we're not using our sites properly. And then if you look at the ESF study, I found that the technology doesn't yet exist for this. They said not released 0.05 microns under any circumstances. I found there were several papers looking at filtering out viruses out of water, compressed water, and they had approaching 100% success, and one paper achieved 100% success. It used an experimental filter using carbon nanotubes, which is a discovery from a couple of decades ago or something, I can't remember, but I remember it was quite news when they found them, but the carbon nanotubes, and they used them doped with silver, and that only works to filter out particles from compressed water, and that's its obvious maintenance issues. And so my conclusion is that this NEPA EIS has to be stopped. It's not fit for purpose. And more than that, NASA need to make sure that this debacle doesn't happen again. They could respond by doing a sterilized sample return, which retained much of the interest, and the other possibility is a new size limits review, which might reduce the size limit to 0.01 microns if it decides fibrocels are now plausible. And whatever the size limit is, we need some idea of the feasibility of it, whether it can even be done, the cost of it, and the time scale, given it's not been done in a decade. So it's only going to happen, I think, if people don't need this level of containment. So there's very little research into it. And they need, especially if it goes down to 0.01 microns, nobody needs that. The 0.05 is very roughly needed for pollution in certain connections. It could be useful, but there's not a big demand for it. NASA will need to create the demand, develop the technology, and then work out how long it's going to take to develop this technology. And they need some idea of the feasibility and cost and timescale. It would be a major NASA project to develop the technology, and they would need to start right away. Indeed, they should have started a decade ago, if that was really what their plan was. So this is not a reason to be scared, because the risk is probably not high. It's mainly geological specimens. They're not looking for life. Life is rare on Mars, even with the highest estimates, because it's so hostile there. It's probably at its highest, it's probably at the lowest end of our coldest, driest deserts, where you go up to maybe a few thousand per gram of the cells, and very unlikely it's as high as that in Jezero crater. So we're looking at tiny amounts of life, but it's not zero, but they're not looking for it. So the example I use is, it's like if you're building a house, then you don't panic about fire, you don't expect fire, but you have your smoke alarm just in case of this remote case, like this fire where it was sent on fire and they were saved by the smoke alarm. And this is a smoke alarm for the entire Earth, with billions of people, and they have had no say in this, and they have had no choice in this, and they should have this say in this. And the Mars sample return studies say that from both an ethical and a legal point of view, that it's not permitted to do it, and it's also ethically wrong to do it without the rest of the world having a say into whether they're going to take this even very small but not demonstrably zero risk. And especially if you're considering this the first of probably many samples returned from Mars, and others will return samples that are much more likely to have life in them. So we need to set the right precedent. So we need to do this properly. And NASA is other countries look up to them and just listen to what they say. Now, I want to talk about how this went wrong. So if I such a page for, this is Margaret Race. This is how I think it went wrong. So how did this happen? And so Margaret Race of the SETI Institute, she says that scientists are likely to focus on technical details, mission requirements, engineering details, and cost of the space operations and hardware. And general public, and this was way back in 1996, she wasn't looking at it, she wasn't doing this in response to what they've done. And the general public like response on risk and accidents, whether NASA or the institutes can be trusted, worst case scenarios, and the methods of handling and containment, whether they're adequate. And that's exactly what's happened here. So I don't think there's any malice here. I don't think there's any conspiracy here. I think it's just sloppy engineers who have been asked to do a job that is not something they're trained in. And they just think, well, we've got to get this EIS out of the way, we'll slap something together, I'm sure that'll do, add a few sites, and that's that's the impression you get from it. I'm not trying to understand how it happened, that's for NASA to investigate. But and this is also in the way they're handling this. Just about every public response to this has been test first or don't return. And so I now want to talk about how there's something that NASA are doing that puts them in great jeopardy with this. And this is if I go and look here, that's their need and purpose. They really shouldn't have done this, their purpose and need section. This is illegal. This will by itself will strike down the case, I believe. I'm not a lawyer and not an expert, so I do want to qualify with that. But it seems pretty obvious it will. So NASA's environmental impact statement said that they made it as a need that the sample has to be returned the sample return has to return an unsterilized sample so that a terrestrial laboratory can look at it and do a proper and comprehensive sample safety assessment. And that's in their need. It's not in their proposed action, it's in their need for the entire mission. They're saying that this is a requirement of the mission. That narrowly defined. Then if you look at the alternatives, they rule out alternatives on the basis that they must be able to accommodate equipment to do a properly rigorous assessment of the biological safety of the samples. This means this is a very narrowly contrived need, which means that it rules out other very reasonable alternatives. In particular, it rules out a sterilized sample return. So, because a sterilized sample return, there's no risk to Earth, it's 100% safe to Earth, but there's no need to do a safety assessment, but obviously you can't do a safety assessment based on a sterilized sample return. So, but that is in their needs section, so they can't look at a sterilized sample return. And this is illegal in the United States. It's decided in the US courts, Supreme Court, 7th Circuit case, I think it was 1996, I'm not sure, 1990s, and it said it is contrary to NEPA for agencies, so it's against the NEPA process to contrive a purpose so standard as to define competing reasonable alternatives out of consideration, and a sterilized sample return is a reasonable alternative. In this particular case, the aim was the engineers wanted to construct a reservoir, and their need statement said it had to be a single reservoir, so they didn't look at multiple reservoirs. And even though the single reservoir might have been the best solution, because they didn't even consider the multiple reservoirs based on that, which seems like a technicality, but the entire project was thrown out because they'd done something illegal. And so they didn't build the reservoir. They couldn't build the reservoir because they'd done a badly written EIS. So that's the worst case scenario. The justice could say you can't return these samples because because of your EIS. So I suppose they try again, but I mean I'm not sure what happens legally there in that situation. So I'm thinking that I'm expecting NASA to just not respond to my comments. I've made 2 comments so far, another very strongly worded comment saying you just shouldn't do this, but I'm not expecting them to respond to that because I think I'm talking to engineers who just don't understand what it's all about, just don't understand why people would consider this to be important, because they're so focused on engineering and cost. And if you're just talking to engineers, you're never going to make progress. So it might be. So I'm looking at other ways. I'm trying to contact the astrobiologists. I haven't done that yet, and the planetary protection officers and the authors of the obvious thing is to contact the authors of the European Space Foundation study, and get their feedback on this, and then go from there. And I'm hoping that some of them will sign a letter asking NASA to stop the CIS because it's clearly mistaken. And then if, but just in case not, then I think it might be necessary to take it to a legal case if they don't respond to letters and they don't respond to this approach that I'm trying. And because it's very much on the radar, hardly anyone seems to have even noticed this yet. Just a few people who've commented. Do comment on it if you to get more attention, and do share awareness of what's going on. So the so NASA's so that's the NASA needs and purpose. Now I'm just going to explain about they couldn't even they've made it a need to prove the samples are safe on Earth, but they couldn't even do that. They don't explain how they're supposed to do that, and I've seen no paper that explains that this has given an explanation of how to do that, except for early ones using out-of-date science from the Apollo era. And the Apollo era has never gone peer review. It was never went through peer review. That was before NEPA. It was just published on the date of launch of Apollo 11. But they have decided to launch the sample tubes to Mars with a permitted level of biosignatures of 0.7 nanograms per biosignature. And that sounds very little for an engineer and for a geologist, but for an astrobiologist, that means completely filling 7,000 ultramicrobacteria. They're only 0.1 cubic microns maximum, completely with that biosignature. 7,000 ultramicrobacteria volumes packed full solid with just glycine or just DNA. And that's how much organics, contamination they've sent to Mars, which they thought was adequate. They thought that was an adequate reduction. And clearly hadn't had astrobiologists that weren't consulting astrobiologists on that matter. And the engineers, they didn't add sterilization because they're always trying to cut out anything that could be mission critical. And sterilization, if it was completely sterile, it would be in a container which would then have to be opened on Mars, and if it didn't open successfully, then they wouldn't be able to use the sample tubes. So that's why they did that. So this means that that if the tubes were returned unsterilized and they try to do a safety assessment, then they can't do it by detecting biosignatures. They will find huge amounts of false positive biosignatures in there in every single sample tube. And they can't do it by cultivating the microbes, because the microbes, they may depend on other microbes in a biofilm. Others have very long generation times, and this is very common in terrestrial life that's been in very cold surroundings, such as in it's very, very cold on Mars. It gets warm in the daytime, it's often cold at night. So we expect very long generation times of Martian microbes. And it's typical for the generation time to be 6 months or more. So you return a microbe to your lab, you try to cultivate it, and 6 months before your one microbe has turned into 2 microbes. It's that difficult to find life on Mars, on Earth, in these very cold, to prove that it's still alive, and to prove it's still viable. And some of the very old microbes, they don't know if it's alive. It shows some biological activity, and it could have a generation time as slow as 1,000 years. So that certainly seems very plausible on Mars, that there could be microbes there with which take between several months to a year to a decade to maybe 1,000 years before they double, and even return to warm conditions. And there are microbes that can survive in warm conditions, but they have adjusted, they're maybe expressing the DNA in such a way that they are ready for very cold conditions. When they return to very warm conditions in the laboratory, then just you kept warm for a long time, they're still very slow at replicating. And so if there was contamination in the sample, then would likely, even if it was released from the sample in the wild, it would be really quite likely that it would be a couple of years before you've got 2 microbes when 1 microbe was released in the wild. and then you have 4, and then you have 10, and then you have 100, and you have 1,000, and then you have a million, and then they're spreading widely because they get used to the warmer conditions. So that is be very difficult to prove that safe. And then otherwise, others also just can't be cultivated in the laboratory, and this is very common, especially ones that are used to nutrient-poor conditions, and the Mars microbes would be used to nutrient-poor conditions. They just can't manage the very rich cultures that are used in laboratory experiments, and they can't duplicate. It just takes too difficult to duplicate the conditions that they do find habitable. So it's going to be impossible to test it by trying to replicate it. And then if I go and look at the section of the ribosomes, yes, here we go. Is a section of ribosomes. Yes, here we go. I don't actually talk much about the ribosomes here. Maybe it's in the... Oh yes, I covered in my reprint that the persevering cleanroom samples turned up many species only known by the ribosome. That's absolutely common, and included 4 that had a ribosome that didn't resemble any known terrestrial species. And that's not unusual. And that was in the swabs of the cleanroom that was actually used to assemble perseverance. So we can be pretty certain that as they start very carefully analysing the organics returned in the sample tubes, they're going to find ribosomes. And these are microbes that are only known by a tiny fragment of the RNA. Remember, ribosomes are a mix of RNA and protein. And that little bit of RNA is sometimes the only thing that you know about a whole entire genera of microbes. So the vast numbers of microbes that we only know by a tiny fragment of RNA in the ribosome, which happens to be quite easy to study in the laboratory and very stable. And it doesn't change very much, and so it helps you to identify different species by just looking at little bit of the ribosome. And the 4 that had ribosomes that didn't resemble any known terrestrial species, they weren't even didn't even closely resemble any known terrestrial species. So we're going to find that for sure in the samples because they're there because they were sent there from Earth, and then they're just going to be returned again, and then we'll find them in the laboratories on Earth. So there's no way that astrobiologists are going to be able to certify these samples as safe. And and they can't test them by so that entire thing, the entire need and purpose thing, it just doesn't make sense. They haven't cited it, they haven't checked it, and it's illegal to do. Now when you scope it like that, it's just not going to work anyway. So that's another reason why we have to cancel this EIS. It's just not fit for purpose, it's just not going to work. And then what we should do instead, well, the alternative actions should surely include a sterilised sample return. The design decisions were based on engineering and geology rather than astrobiology. They're clear that if they got consultation from astrobiologists, they didn't listen to what they said. They were only listening to the engineering and geology, because this is not fit for purpose for study for looking for life on Mars. It's just not going to work. The past life is too degraded, and the present life is likely to be very rare, and there's so much contamination, unless it's something very obvious like mirror life, they're not even going to be able to see it. And even that will be difficult to detect, because it's difficult to detect things that you don't know what they're like, and you're not specifically looking for. So they'll detect. The first thing they'll detect is terrestrial life, and that's probably the only thing they'll detect. They get very strong positive signal from that, and they won't be able to do any astrobiology. So it's just going to be another mission in finding out that Mars was habitable, which we already know. Jezero Crater was very habitable, it had a delta. They learn a little bit about the conditions in Jezero Crater at the time that the delta was formed, and that's about, and then it will be a technology demo for astrobiology. It's shown that we can return samples, and so what will be important will be the future samples we return. These samples are not important for astrobiology. And so but it could be made into a mission of much greater astrobiological interest by sending sterile containers. That will turn it into something that is of almost no astrobiological interest, to something that has some preliminary astrobiological interest, some quite significant preliminary astrobiological interest. And there are two things. There's one thing that is not much talked about with, indeed, I think it's a thing that I'm paying attention to with my preprint, and that is that that we should be interested in the chemistry of planets that don't have biology on them. Even if we find habitats on Mars that are uninhabited, and this is of great interest, not just for saying that, not just for trying to home in where the life is, we're not just interested in habitats, but the very few uninhabited habitats on Earth. When you get a meteorite, when you get a volcanic eruption, and for a little while the basalt ejected from the volcanoes hasn't been colonized by life yet, and that's about the closest we've got to uninhabited habitats on Earth. But this may be the most common kind of habitat there is in the universe, and the only place we can study them is on Mars. There might be uninhabited habitats on Europa and other places, but certainly the only terrestrial uninhabited habitats that we can study are on Mars. And this is very important for prebiotic chemistry. And we're only really going to be able to understand the origins of life reasonably well by having a good understanding of what chemistry was like before life. So if Mars has no life on it, it is very precious. And we can look at it's got extraordinary range of habitats. It's got these deep salty lakes way below the ice caps. And we can look and find out what the organics do in a salty lake that has never seen life. We can look at the surface chemistry of the dusts and the salty brines and things without any life. And even astrobiologists don't seem to have picked up on this, even though they talk about these topics. This is where I come across this with a different perspective, because I've lived through a lot of these subjects just developing from scratch when I was a kid. I've been very interested in a very broad range of scientific topics and followed them keenly for decades. And so I can do a literature survey of a very wide range of different topics, and that is the main thing that I bring to the topic. I'm not a specialist in astrobiology as such, but I can read the science in different areas of the topic. And I've got a very strong maths background. That's how I did my degree in maths. Astrobiology is a very multidisciplinary subject. A very interesting subject because it has no examples of its subject matter yet. And so astrobiology, it's the Viking mission has studied the surface of Mars, and the Viking labeled release turned up very intriguing results. And this should be people saying, well, they shouldn't. And then the reaction has been from the geologists and the general scientists has been, you didn't prove there's life there. There surely isn't life there. It looks very sterile. It's just not interesting. But for astrobiologists, these experiments should be interesting because they found either biology or they found very interesting chemistry. The surface now seems to be much more habitable than it seemed at the time of Viking, with the discovery of the salts and the liquid and the brines. Curiosity found very cold but brines that form every evening and every morning in Jezero crater, in Gale crater, and the same brines that's with less than 100% humidity. Jezero gets 100% humidity, so it's more likely to have these brines. It's slightly lower and it's got higher humidity and slightly colder. And by studying these, Viking found that when the temperatures peaked, because the natural daylight circles, it wasn't done deliberately, it was an accidental experiment. You got the day-night changes in temperature, and when the temperatures are high, then more gases were evolved in the labeled release, where they detect evolved gases by the radioactive signature of the radioactive organics that they fed whatever was in the dirt. And this was released 2 hours after the peak of the warmth. With chemical reactions, you can explain 5 or 10 minutes delay, but the 2 hours delay is, and this is Joseph Miller who says this is a clear signature. This would be regarded as proof that there's life here if it was on, if we found that on Earth. We don't know a way of doing this with chemistry. So there's either very complex, difficult to understand chemistry, and it could be that very complex prebiotic chemistry can do that, or it is actual life. So we need to find out what it is. It's interesting for understanding prebiotic chemistry. It's also going to help us to deal with searching for life on Mars, because we will know what to look for and what not to look for. So we need to understand, and this shouldn't just be treated, oh well, we don't understand this, but it can't be life. We need to understand things that aren't life on Mars, because they're interested in prebiotic chemistry, and because they help us to look for life as not being these things. So this can be made into a far, far more interesting mission, and probably not at much by way of upfront costs. So first of all, we've got to return these extra samples. So that means slightly modifying. So they're going to have to send arms to pick up the sample tubes and bring them into, and put them into the sample container, pick up sample tubes from the ground and put them into these orbital returns as sample return capsule. And this is not that different from Viking, which picks up a scoop of dirt from the ground and puts it into the containers inside Viking to do the experiments. And so I'm suggesting that we either have a multi-purpose, either have 2 attachments, or we maybe even just make one of the things that make the pincers to dual purpose as a scoop. It's got a little scoop attachment to it, and it scoops up some dust. And I'm sure engineers can find ingenious ways of making it so it can both pick up sample tubes and scoop up dirt. And so it scoops up some dirt and it puts it in a sterilized container. And the container would be sterilized on Earth, and it could only be opened on Mars. And this would be mission critical for that particular experiment of picking up dirt and putting it in a sterile container. But astrobiologists would see that as being something that is worth taking that risk. So, but it wouldn't have any effects on returning these sample tubes. They can still be returned as normal. And the digging would be done at the end, so if you're worried about that damaging the digging arm, then that can be the last thing it does. And if something goes wrong with the digging, then it just returns anyway, and it just returns the samples without the dirt. So this is a bonus sample. It's not going to return much expense at all in modifying the SF sample returner and adding this extra capsule, this extra container to the capsule. And yeah, it'll be hugely interesting for astrobiology. And then the other thing is to I'll show it just briefly. It's going to return. We've got these little compressors, and you've got MOXIE on Perseverance, and it will compress the atmosphere on Mars to its Earth's pressure. And you'll be able to get, if they can have a 1 litre container, it will return about 0.2 grams of Martian atmosphere. And in the process of compressing, and after compressing, and then in a sterile container, and with a getter first to remove evolved gases, and then they do a cycle which then collects dust from the dust storms, and so that will collect up to several grams of dust, and the atmosphere can be analysed much more precisely, so we get a very clear idea of what the atmosphere is like. And we can do experiments with the dirt and with the atmosphere, and we can find out what they're made up of, and we can do analog dirt and analog atmosphere on Earth, and we can do much more accurate simulations of the Martian surface with these results as well. So this will hugely increase the interest of the mission, especially for astrobiology, for geology too, for safety assessments for astronauts. If we ever can send astronauts to Mars, then all this is going to help with that. It's not a big increase in costs, so I'm going to explain then how we can return safely. And the compressor is something that a very major compressor it can fit inside a sample tube. So you're not talking about something big. You're not talking about something heavy. And then it's used to collect a lot of gas and a lot of dust. And so this is how we can do it. So the solution to is you check for life first, and then return the sample to a safe orbit above GEO to study remotely with miniature instruments. And they can check the dust, and if it is possible to check for life, then these sterile samples returned in sterile containers are ones that we could test. Though I'm not sure, we have to look very carefully about whether we can get reasonably good proof that there isn't any life in them if we want to return them unsterilized. But what we can do, we can send huge things up to geostationary Earth orbit. I worked out that geostationary Earth orbit wouldn't actually be that orbit. It would be about 10, 20, 30,000 kilometers above it. Still very far in delta V from the moon and Earth. No risk to our biosphere, and not actually in the plane of Earth's equator, but slightly offset in a plane called the Laplace plane. And this is a situation where even if the satellite explodes, then the debris and the dust and anything else, it's the balance of the pressures of the light and the gravity of Earth keeps them stabilized in the ring plane. It's the plane where Earth would have rings, where ring particles would orbit if Earth had rings. This is very, very safe. There's no possibility of material, even in the event of an explosion of the facility, of it contaminating Earth's biosphere. Very, very safe place to return to. And then we already have the capability to send 9.6 tons in one mission up to above geostationary Earth orbit with the Ariane 5 spacecraft from ESA. So we can send huge things, and the astrobiologists, they have most extraordinary instruments that they've designed. They can now put, and I remember when I was young in my 70s, that an electron microscope would fill an entire room. And now they've got a miniature electron microscope which can which they could send to Mars. Just, I think it's a fraction of a kilogram. It's not very big. And the EIS, the NASA EIS, claimed that we have to return them because we've got to do sample preparation. Well, the astrobiologists, of course, have thought about sample preparation. That is included in their instruments. They can detect a single amino acid in a gram of sample, and they've got instruments they could send to Mars that does all the sample preparation, and it has these miniature, they use microfluidics to move things around in tiny, tiny labs on a chip. And they can do everything without humans there. So you don't need to send humans up to look at it. We don't need to return to terrestrial laboratories. And they can even do gene sequencing. They have end-to-end gene sequencing, which can take a sample, prepare it, analyze it, do all the computation as well, which takes a lot of data. They don't need to return all the data, and then they return the gene sequence to Earth. And that could be done in orbit. It could be done on Mars. So it could certainly be done in orbit. And they can do super resolution optical imagery, which goes beyond 0.2 microns. By the way, if I go up to the top, just in case you don't know, this photo was taken with an electron microscope. We can send miniature electron microscopes to Mars if we wanted to. They could take photos like this. The preparation, everything done properly. And they can also do, there's the idea of, if they do return life, then the Viking experiment, it got this life producing gases by wetting the samples and giving them organics to eat. So we could get little microbes swimming around. Well, someone has built an off-axis holographic imager, and this can look at a sample that's got swimming microbes in it. It can take a snapshot of it, and it's a holographic image, and it can be focused on Earth. You return just the holograph, and then you can focus on anywhere in the 3D plane. you get a sharp, crisp image of the swimming microbe. And it's even better, you don't have to try and follow it or anything. You've got this static image which has got a photo of a microbe in it, and then you can focus it when it gets back to Earth and get a precise, ultra-sharp image of this swimming microbe. And we've also got Raman spectroscopy, where you can actually find out, you can focus it in, so you've got synchronizing optical photographs of something, and then whatever you are focused on, then you can also take a spectrograph of it, or you could take the Raman spectroscopy, where you get an idea of what it's made of. So you can focus that into a very small scale, into just a bit of a microbe, and find out what it's made of. And so that's another thing that they're planning. They have designed instruments that they think can be sent to Europa that can do that. So we can certainly study them in orbit with very, very advanced instruments that we now have. We couldn't have done this a couple of decades ago. In year 2000, it's been utterly impossible. A gene sequencer would fill lots and lots of machines filling a laboratory, but now we can fit them into something you can hold in the palm of your hand. So it's just extraordinary what's happened in biology. The things have been just miniatured so much, and we can send 9.6 tons of this stuff up to orbit in just one mission. So this whole thing that we can't prepare it on Earth, we've got to prepare it on Earth, it's not true. We can prepare it in space, we can do all the studies we need in space, for all the preliminary studies. If we then get to a point where we have something we want to return, then we then need to look at safety assessment. And so now we can't send humans to orbit. So I've got some examples to explain why that's not possible. So if I look up Cinia, this is a simple. Wait a minute. I did two sets by mistake. Right, two Cinia plants on the ISS. And so this is an example that shows that human quarantine can't protect Earth from molds that might impact on our crops. So some of your keen space enthusiasts may remember this incident, where they tried to grow these Cinia flowers, which is an edible flower. They tried to grow it on the ISS, and the two of the plants died, despite all the care they were taking. And they found out it was killed by a mold, which probably got there on an astronaut's microbiome. And this was this mold called Fusarium oxysporum. And an astronaut could have this for weeks, for months, and they just got this mold in the microbiome, and it's not doing any harm to them, and yet they've been up in so there's no quarantine period that can make you safe. And then you come back to Earth, and some of it, and then some of it gets discarded from your skin, and then it lands on a crop, and that crop gets moldy and it starts spreading across Earth, and then it may have some serious agricultural impact, and there's no way you could detect that. So quarantine is not going to work, and there's no way, even if you knew it was there, that you could stop it coming back. So and this particular case also, it's an opportunistic pathogen of humans. So sometimes people are immunocompromised, they can get very sick or even die as a result of this, as an opportunistic pathogen. So that's another example. That's an example of a symptomless spreader, someone who's got a pathogen of humans, but it's not pathogenic to them because most people are not affected. And there's some very clear examples. There's examples of the COVID, of course, it's happened a lot. We've had symptomless spreaders, and neither of these cases seems to be looked at in the planetary protection literature. It's one of these things where, coming at it with a multidisciplinary way, then I spot things that just people haven't even thought of looking at for some reason. But as soon as the World Health Organization gets involved, and once it becomes clear that large-scale effects are not totally ruled out, they will look at it. Then the first thing they will say is, how is quarantine going to protect us from a symptomless super spreader? And they'll have examples like Typhoid Mary, who had typhoid all her life, and when she cooked food, then it killed some of the people that she made food for, and she never believed she actually had typhoid, and was confined to an island for the rest of her life, never felt sick at all. So quarantine couldn't keep out typhoid Mary. And this Legionnaires' disease, which is the closest analogue, is an actual disease of biofilms, and it invades our lungs by the same method it uses to invade the protozoa that graze biofilms, and that's used as an analogue of a disease, an opportunistic pathogen that we might get back from Earth, from Mars. And again, some people get Legionnaires' disease who have no symptoms, and other people die of it. So having no symptoms when you're in quarantine would not mean that you're safe. And there's no quarantine period that can be guaranteed. And then the most impossible of all to try and quarantine against is, suppose you had these ribocells that I talked about. Now suppose they are mirror life. And so now if we go and look at these ribocells again, and go and look up here, these tiny things, and being so small, they are immune from grazing, because the larger, even an ultramicrobacteria would not be much interested in a little globule of food that's just that size. And a great big protozoa or something is not going to be grazing on that. So it helps it against predation, and then it's also very good because if it's in nutrient poor environments, then it's got a much larger surface to area, surface to volume ratio, and this means that if there's a certain maximum rate at which nutrients can be moved across its surface, then it can use up its nutrients much faster. It can take up nutrients much faster, and so it could grow. It wouldn't be able to swim by itself. Brownian motion would just scatter it around all the time. But there are microbes so small that they can't swim really. They just get randomly moved around wherever they go. And you just see them in Brownian motion. They're just jiggling around all the time, and there's not much they can do about deciding where they're going. And so this ribocell thing, then it could be quite competitive on Earth. And suppose now it's mirror biology, and suppose it's got the enzymes to slowly metabolize organics of both chiralities. Or maybe it is even chirality indifferent. That's another possibility, mirror life, or it's mixed chirality. And it just, but yeah, let's keep with mirror life, just keep the one thing. And it's got the enzymes to be able to metabolize both types of organics, because most organics on Mars is not formed by life. Even if it's mirror life, most organics would be a mix of the two, because it comes from meteorites, so it's made locally, because most of the surface Mars is uninhabitable, and even for life like this, and there's a lot of organics there coming from space. So even if you've got microhabitats, most of the stuff around it will be organics, but there won't be anything able to use it. So most of the organics we turn is not going to have life in it, but the life that can, it'll have a very strong selection pressure to be able to eat both types of organics. So the end result of this is that, and there might be bigger ones, there could be ribocells, there could be bigger ones, but the ribocells will obviously be very difficult to keep out, especially difficult. Then gradually, once it's got to Earth, then it just needs to establish a foothold. Then gradually, maybe take decades, may even take centuries, but gradually the mirror life would spread out across Earth's biosphere. There'd be nothing we could do to stop it. It would gradually turn everything into mirror organics. Most terrestrial life would not be able to evolve to use mirror organics, and then humans wouldn't go extinct, as that wouldn't happen, but we would have to gradually cover Earth, terraform it, cover it with greenhouses basically that can keep mirror life out of it, or keep mirror organics, at least keep it under control enough so we can maintain normal habitats inside it. Have to do the same with the seafloor. It might even happen in our generation. It might happen a couple of generations from now. But that is something also that we need to consider. That be turning something that might make Earth very difficult to be significantly less habitable and require quite a lot of technology to live here as a result of some micro. They wouldn't be too pleased that we'd return mirror life in the 21st century, because of all the awkwardness and difficulties and problems it caused for them. So that again is something that we have to be careful about, and you can't stop that with quarantine, because it's very unlikely that just a few microbes in real life, and maybe with maybe it takes years to duplicate, even 1,000 years, and then it's a very far future problem. But if it takes a year to replicate, and you've got some of that in your, or you've got life that replicates a little bit more much, and it's just mixed up and it joins your microbiome, it's still very, very unlikely that you're going to be harmed by it. So this is why we can't send humans up there. And this is not, it's not been through peer review, but I think it's kind of obvious, and I'm not expecting that to fail peer review when eventually I submit to the journals. I'm expecting these points about the synchronous super spreader, about the returning of mirror life. I'm expecting these to pass peer review, because I don't really see how there could be anything, how they can fail. And so to keep Earth 100% with minimal cost, we don't have humans up there, we have them remotely studied, we sterilize the samples returned to Earth, and we sterilize subsamples of the dust and dirt. So now, the way we combine this with the geological samples is that we do split the capsule apart, but we don't split the sample tubes. We just open the lid, whatever it is, take out the dirt, the container of dirt, take out the container with the dust and the atmosphere, and we put those in the thing above geostationary Earth orbit. We would then sterilize the rest with our X-ray emitters or whatever it is that we're using. And in this experiment, we could send up 9 tons of sterilization equipment up there to sterilize it, if that was needed. And then we return the capsules. We don't need to do the nanoscale X-ray emitters for this particular version of it, because it's returning to a satellite above geostationary Earth orbit anyway. A great big heavy one. It could be 9 tons of satellite. And then, having sterilized the samples, we then send that on a spacecraft down to Earth, where it is carefully break the chain of containment of contact with Mars, so it is completely safe. And so that is a way we could do this, which is totally 100% safe for Earth, greatly increase the astrobiological interest, greatly, and it's increased somewhat the geological interest. It's put no extra impact on the geological samples, minimal delay, because it just needs to stay up there long enough to be sterilized, and then it returns. It's just got a very, very tiny few days delay or whatever is needed, depending on what you use to sterilize it. And so that is clearly a reasonable alternative. And I'm not sure that it would even cost NASA more. A lot of the expense would be met by universities, just like they combine together to find the funding to build these big telescopes. So the big telescopes in La Palma, they are not in Hawaii. They are not paid for by NASA. And in the same way, universities would find the funding to send great big experiments to do the astrobiology in space, and NASA wouldn't have to pay for that either. All they'd have to do is to pay to send a small satellite up there, and they would save on the cost of the aeroshell, because they're not using the aeroshell. They'd have a larger payload that they can return from Mars to Earth. Their Earth return sample capsule can be a little bit larger. There are orbits that I looked at, which they're using an ion thruster, which they have as a possible way of returning. Then they can use very efficient orbits that let them get back to Earth using a distant retrograde orbit around the Moon, and using first the Earth Moon L2 and L1 position. They do a spiral in around the and then they do a spiral out on the other side, with very little delta V, less delta V than is needed for a conventional Earth return. Then they could get a larger sample back to Earth without the mass of the aeroshell. So it's actually more return. We actually, if you're worried about the extra mass of the bonus for the astrobiologists, you could actually take quite a lot of extra mass. You could even, just get the ESA rover to pick up a few rocks as well to add to your geological sample. A few little pebbles from the surface that you look at and you say, well, that looks interesting. Pick up one of those as well as the sample tubes and pop them in your now rather larger orbital return capsule. Could be a bit larger as well, I expect, for engineers to look at. So we are looking at or else it's lower cost because you've got a smaller thing to return and you don't have to use your aeroshell. So it's either lower cost or it's a little bit bigger. And so that's it. I think that's a more interesting mission for everyone. And it's certainly a reasonable alternative that should be included in the EIS. So the EIS should include sterilized return and something like this, return to a satellite above that is safe, where you can do the astrobiological thing. So I say that it should include both these possibilities, some variation on them. And the third possibility is where they can return it to Earth. And I look briefly at that in my preprint as well. And a safe return to Earth, I work out a method using a sump instead of an airlock to protect the and inside a nuclear bunker and returning in a titanium sphere. And if you combine all those together, you can work out a way that on paper at least looks like it could be 100% way of returning unsterilized samples to Earth, and then observed and then looked at telerobotically. Anyway, you can look at I talk a bit about this on this page. So we have all these ideas that can be looked at, and so that's totally safe. Oh yes, that's my. So that's the example, a little bit of a gap that seems to be at the top of this sterile flow container. So now let's just go up and finish my introduction. I'm not going to do the whole thing, lots of things here. So the two alternatives which both keep Earth completely safe, and they can be achieved with a simple environmental impact statement. They don't need to do a size limits review. They don't need to do very much at all, except look carefully at the method of sterilizing the samples. And then so then the last thing is do comment on the proposal yourself. So they aren't getting many public comments. I'm going to I'll do a link to this video, as I think probably as one of my comments, because it may be of interest to them. And I am confident they are going to re-examine this and do the proper procedures to protect Earth. I do what I can to make sure it happens sooner rather than later, because it's going to be a big expense, and it'd be a terrible thing if they return the sample, if the public only realise when the samples are on their way back from Mars to Earth. There's a big hoo-ha, and then the president says, well, you just got to divert your satellite so it avoids Earth. And that would be awful. So from the point of view of science return. So I'm doing what I can to make sure that it all happens sooner rather than later, because they're going to face this at some point. And the sooner they face it, the better it is. And the better the mission is going to be, and the lower cost it's going to be. The more preparation they can do, they can make it a much better design. They've got much more time to work on the changes they're going to have to make to the mission in order to make this work. Because all the methods are going to involve some change, including a sterilized return. And so I'm confident this is going to be done. And then this is going to be a good precedent for other countries that don't have the time that NASA has and don't have the resources they have, and they just want to return a sample quick and dirty sample returned from Mars. Then they can do a sterilized sample return. And that will become the norm. And if you want kudos that, oh, I've returned a sample from Mars, then you can just return a sterilized sample. And lots of countries can do that, and sterilized samples will teach us a lot about Mars, especially if they return them inside sterile containers as well. Then you could recognize life in a sterilized sample returned in a sterilized container. You could, if you don't want to do all this elaborate above geostationary Earth orbit, you can also do the astrobiology samples and then sterilize them. That might seem rather pointless, but it isn't. if you were to take these things, these samples of dirt and samples of dust and dust storms, and you sterilize them, that does not mean erasing all the organics. And especially present-day life would be easily recognizable in a sample that is taken in a sterilized container. So that is also an alternative for the astrobiological samples, that you return them to Earth completely sterilized. And if you think there's a very low chance of life in them, then that's maybe that you decide that's what you do. And you say, well, we'll just sterilize them, we'll still be able to recognize, you'd recognize chlorophyll, you'd recognize any complex molecules, you recognize pretty well, you'd recognize DNA, we'd have quite a few breaks in it, but it'd be obvious that it was DNA. You'd recognize the proteins, you'd recognize that there's a chiral imbalance to the amino acids, and you could do that with a level of sterilization that would still mean that it's completely killed. So that also is a possibility if you think there's a low chance of viable life and you don't want to try growing it in space. So anyway, do click on the blue comment button to click there, and then click on the blue comment button and whatever your own views are. And I hope that by doing this, I just happened to be in the right place at the right time, and so not many people have the right background to do this careful check of the environmental impact statement, and those who do have the capability, I'm sure, have lots of academic work they have to do, people to teach, they have projects they're doing, they have research commitments, and so they just haven't had the time to look at this properly. And I've been lucky enough to be able to spend quite a few hours working, quite a bit of time indeed. I've spent pretty much all my spare time on it for a few weeks, and I could have mentioned this right when I did my first comment, beginning of early in November, but I want to be absolutely sure that I'm not missing anything. It's quite a big thing to say, but I mean it's absolutely clear. I don't see how there's any possibility that this is wrong. That I've probably made mistakes. Please let me know if you notice any mistakes. But the broad picture is absolutely clear. There's just no way that them not mentioning the ESF study, that can't be that I've misunderstood something. Surely, I mean, do please explain if I got something seriously wrong and there's some reason why they shouldn't mention the ESF study, and do let me know. I mean, that would be very embarrassing, but I won't be embarrassed if they've got anything wrong. Do let me know, or if there's some reason why this is actually a site that does support the existing credible evidence that the conditions have not been amenable to supporting life. But I mean, it doesn't make the blindest bit of sense. So anyway, so that's it, and that's what I'm just doing. You just do what you can. I just want I want Earth to be protected, and I want the science to be good, and combination of both two. And I see no reason why we shouldn't do both. Good science, safe Earth. Why not? What's the point in unsafe Earth and bad science? Which is basically what they're doing. So please, safe Earth and good science. That's basically it. And let this be a good mission. So that's what I'm trying to hoping that people will be able to achieve.